Unraveling the mystery, the intersection of bioelectricity and RF radiation. Bioelectricity, RF radiation, and the paradigm shift in understanding health risks. Recent advancements in RF radiation research, including DARPA's RadioBio initiative and FDA-approved treatments like TheraBionic, along with the success of the Oncomagnetic device in treating gliomas, are significantly altering our perception of non-ionizing radiation's effects. TheraBionic, for instance, utilizes RF radiation at power levels up to 1,000 times lower than cell phones to effectively treat inoperable liver cancer through non-thermal means. Similarly, the Oncomagnetic device employs spinning oscillating magnetic fields, or SOMEF, to induce cancer cell death via oxidative stress, which is also the mechanism that potentially damages healthy cells from cell phone radiation, thus representing a non-invasive and potentially superior cancer treatment approach, when harm to healthy cells is prevented through selective frequencies and exposures. These advancements demonstrate biological effects, including changes in DNA methylation and gene expression at non-heating radiation levels, challenging the long-standing belief that non-ionizing radiation's primary impact is thermal. This shift in understanding highlights the potential non-thermal, epigenetic impacts of RF radiation, marking a pivotal moment in our grasp of its biological implications. Current understanding of non-ionizing radiation. The traditional view of non-ionizing radiation, such as the type emitted by cell phones and wireless devices, has been largely based on its thermal effects. It was long believed that the primary concern with non-ionizing radiation was its capacity to heat biological tissue, akin to how a microwave heats food. This perspective underpinned safety standards and regulatory guidelines for decades, focusing primarily on avoiding excessive heat generation. However, the distinction between thermal and non-thermal effects has become a central point of debate in recent years. Non-thermal effects refer to biological changes caused by electromagnetic fields at energy levels too low to cause significant heating. While the thermal effects are well understood and relatively straightforward to measure, non-thermal effects are more elusive, complex, and controversial. Recent advancements in RF radiation research are pivotal in this debate. DARPA's RadioBio initiative, for instance, aims to understand how living cells respond to low-level RF radiation, suggesting that there could be fundamental biological processes affected by RF energies lower than those traditionally deemed harmful. Similarly, treatments like TheraBionic, which use RF radiation to treat inoperable liver cancer, operate at power levels that are orders of magnitude lower than cell phones. This treatment, along with the Oncomagnetic device's success in treating gliomas through spinning oscillating magnetic fields, demonstrates significant non-thermal interactions at the cellular or molecular level. These advancements challenge the traditional paradigm by demonstrating that non-ionizing radiation can have profound biological effects without necessarily causing a significant increase in temperature. The implications of these findings extend far beyond the realm of medical treatment, prompting a reevaluation of our understanding of cell phone radiation and its potential health risks. Revisiting major studies on cell phone radiation risks. Key studies have played a crucial role in shaping our understanding of cell phone radiation risks. The Interphone study, Hardell Group studies, Serenat study, U.S. National Toxicology Program, Ramazzini Institute study, Reflex Project, and the Bio-Initiative report collectively suggest an increased health risk from cell phone-level electromagnetic radiation. These studies have examined various outcomes, from brain cancer risks to changes in brain activity and sleep patterns. The methodologies of these studies range from large-scale epidemiological research to controlled laboratory experiments. For example, the NTP study, one of the most comprehensive assessments of health effects in animals exposed to radiofrequency radiation, found evidence of carcinogenic activity in rats. On the other hand, the REFLEX project explored cellular responses to RF radiation at the molecular level. While these studies collectively point toward potential risks, they have also faced criticisms regarding their methodologies, interpretations, and reproducibility. Some argue that these studies often struggle with confounding factors, such as the variations in individual usage patterns and the rapid evolution of technology, which can affect the relevance and applicability of the findings. Connecting the dots from cancer treatment to cell phone safety. The way RF radiation is used in treatments like TheraBionic and the Oncomagnetic device offers insights into how cell phone radiation might affect human health. TheraBionic's use of RF radiation to treat liver cancer and the Oncomagnetic device's treatment of gliomas through induced oxidative stress demonstrate that RF radiation can interact with biological tissues in complex ways, beyond mere heating. These treatments raise questions about the potential biological mechanisms through which cell phone radiation might pose health risks. For example, if RF radiation can disrupt cellular signaling or modulate the immune system in a therapeutic context, it's plausible that similar mechanisms could be at play in scenarios of prolonged exposure to cell phone radiation. Understanding these mechanisms is crucial for assessing the risks associated with everyday exposure to RF radiation from wireless devices. The role of non-thermal interactions in health risks is at the forefront of current research. Studies indicate that radiofrequency radiation can cause resonance effects, disrupt cellular signaling, and even modulate the immune system without causing significant heating. This opens up a new dimension in understanding how cell phone radiation might impact health. Research into DNA methylation and gene expression changes due to RF radiation further supports the idea of significant biological effects. These epigenetic changes could have long-term implications for cellular function and disease susceptibility. Understanding how RF radiation influences these molecular pathways is key to comprehending the broader health implications of prolonged exposure to cell phone radiation. Currently, guidelines and safety standards for cell phone radiation are based largely on avoiding thermal effects. However, with new research findings suggesting significant non-thermal effects, there is a growing call for these guidelines to be reassessed. The potential implications for public health policies are significant, as current exposure limits may not adequately account for the non-thermal interactions now coming to light. This could lead to changes in cell phone usage guidelines, manufacturing standards, and public health recommendations. Policymakers and regulatory bodies will need to balance the benefits of wireless technology with the potential risks to health, possibly leading to a redefinition of what constitutes safe levels of exposure. There are still many gaps in our understanding of the health impacts of cell phone-level radiofrequency radiation. Future research needs to address these gaps through well-designed studies that can isolate the specific effects of RF radiation from confounding factors. Interdisciplinary research combining medicine, biology, and physics will be crucial in advancing our understanding of these complex interactions. Studies should aim to replicate previous findings, explore new potential health risks, and develop a more comprehensive understanding of the mechanisms at play. This will not only help in refining safety standards, but also in harnessing RF radiation's potential for therapeutic purposes. Insights from Michael Levin's research on bioelectricity and collective intelligence. The paradigm shift in biomedicine. Michael Levin's work challenges the traditional biomedical paradigm that focuses primarily on genetic components and biochemical dynamics. This narrow approach limits interventions to micromanaging cellular components, often addressing symptoms rather than providing permanent solutions. Levin introduces the concept of diverse intelligence, which examines the goal-oriented capacities of various systems and suggests a paradigm shift towards understanding cells and tissues as collective intelligences navigating problem spaces. Bioelectric networks, which coordinate large-scale anatomical goals, offer a tractable interface for biomedical interventions. Core concepts. Limitations of current biomedicine. Current methods focus on molecular-level control, which is inadequate for true regenerative medicine. Existing interventions often address symptoms rather than providing permanent repair. Anatomical compiler vision. Levin envisions an anatomical compiler that can design and stimulate cells to form desired structures by leveraging the goal-seeking behavior of cellular networks. Bioelectric networks. Bioelectric signals act as a cognitive glue, binding cells into coherent structures and enabling complex organogenesis. These networks facilitate top-down control and coordination across cellular collectives, essential for large-scale anatomical goals. Collective intelligence of cells. Cells possess proto-cognitive abilities, such as memory and decision-making, even at the subcellular level. Tissue-level intelligence can be harnessed for regenerative medicine and bioengineering. Cancer and collective intelligence. Cancer can be viewed as a failure of the collective intelligence of cells, where individual cells revert to more primitive, self-centered behaviors. Modulating bioelectric signals offers potential pathways for cancer treatment by restoring collective cellular goals. Research programs and applications. Levin proposes research programs that leverage bioelectric and cognitive science tools for biomedical interventions, aiming to collaborate with cellular intelligence rather than forcing specific outcomes through molecular manipulation. Mitigating entropic waste for optimal therapeutic outcomes. Entropic waste refers to the disruptive and disorderly impact of radiofrequency radiation on biological systems and natural environments. It encompasses the non-thermal, often invisible effects of electromagnetic fields that contribute to biological stress, environmental degradation, and a decline in the health integrity of exposed organisms. Impact of entropic waste on bioelectricity. Bioelectric dissonance. Entropic waste can cause bioelectric dissonance, disrupting the natural bioelectric signals that coordinate cellular and tissue-level activities. This dissonance undermines the coherence and functionality of bioelectric networks, leading to impaired cellular communication and coordination. Disruption of homeostasis. Bioelectric dissonance caused by entropic waste can disrupt cellular homeostasis, affecting processes such as cell proliferation, differentiation, and migration. This disruption can contribute to various health issues, including cancer, neurological disorders, and developmental abnormalities. Importance of mitigating entropic waste. Restoring bioelectric harmony. To achieve optimal therapeutic outcomes, it is crucial to mitigate the effects of entropic waste and restore bioelectric harmony. This involves reducing exposure to radiofrequency radiation and other sources of electromagnetic pollution. Supporting cellular intelligence. Mitigating entropic waste supports the natural goal-seeking behavior and collective intelligence of cells, enhancing their ability to repair and regenerate tissues effectively. Enhancing regenerative medicine. By minimizing bioelectric dissonance, we can enhance the efficacy of regenerative medicine techniques that rely on bioelectric signals to guide tissue formation and repair. Conclusion. Michael Levin's groundbreaking work emphasizes the importance of understanding and leveraging the collective intelligence and bioelectric networks of cells for next-generation biomedicine. Addressing the impact of entropic waste on bioelectricity is essential for optimizing therapeutic outcomes and advancing regenerative medicine. By mitigating bioelectric dissonance and supporting the natural competencies of cells, we can pave the way for more effective and sustainable biomedical interventions. Abstract. Recent advancements in radiofrequency radiation research, particularly DARPA's RadioBio initiative and innovative treatments like TheraBionic, along with the Oncomagnetic device's efficacy in treating gliomas, are transforming our understanding of non-ionizing radiation's biological effects. These groundbreaking studies and treatments indicate that non-thermal interactions, such as resonance effects, cellular signaling disruption, and immune system modulation can occur at radiation levels significantly lower than those emitted by cell phones. This evolving understanding challenges the long-standing belief that the primary impact of non-ionizing radiation is thermal. In this context, it's crucial to revisit and reassess the body of research indicating potential health risks associated with cell phone-level electromagnetic radiation, including major studies like the Interphone study, the Hardell Group studies, and the U.S. National Toxicology Program findings. By addressing the impact of entropic waste on bioelectricity, we can optimize therapeutic outcomes and enhance our understanding of how to mitigate the potential health risks associated with prolonged radiofrequency radiation exposure.